title of my talk is The Role of Clinical Exome Sequencing in the Genome World. And I'll be talking a little bit about our experiences using the Twist exome and specifically some customizations we've done to the Twist exome to sort of better close the gap with whole genome sequencing, and a bit of a discussion about what that, why we've done that, and what advantages that provides us in our setting here. But firstly, a bit of an introduction as to who we are. So VCGS, the Victorian Clinical Genetics Services, is a not-for-profit subsidiary of the Murdoch Children's Research Institute. We're located at the Royal Children's Hospital in Melbourne, Victoria, Australia. We have a comprehensive laboratory and clinical genetics service. And we have, because we're attached to the Murdoch Children's Research Institute, we play key roles in research and development, and particularly in the translation of research into the clinic. We offer testing throughout all stages of life, genetic testing throughout all stages of life, from the preconception and pre-pregnancy stages through our carrier screening tests, into the prenatal period with our NIPT and prenatal testing using microarray and exome and genome sequencing. Newborn, in the newborn period, we offer newborn blood spot screening, metabolic testing, and also our ultra rapid whole genome sequencing service, or acute care genomics, which is aimed at testing critically unwell newborns or other children with a rapid turnaround whole genome sequencing. We do a lot of testing in the childhood development phase for Fragile X syndrome through our chromosome microarray service, and also, again, our whole genome and whole exome service, and up to the adult stages of life. So we do offer testing for adults with genetic diseases as well. We have a fairly multidisciplinary approach here at VCGS. We're lucky enough to have quite a lot of support, not just scientific support, but also clinical support and liaison support through teams like our clinical liaison team, who interface with referrers directly at the pre-test and post-test process. Clinical geneticists, we're lucky enough to have some clinical geneticists that are embedded in the laboratory to help us with clinical questions. We work closely with our counselors. We have a team of clinical bioinformaticians that develop our pipelines and various pieces of automation. Obviously, our medical scientists are a key part of the service. And also we, again, work very closely with our research partners. And again, just emphasizing that one of the things that I think sets us apart, or that we try and emphasize, is that we are tightly integrated with the research side of MCRI. And so we've got, there's quite a lot of work that goes on at VCGS that is sort of translational research, or that contributes to furthering the technology or the field of diagnostic genomics. And this is just a small collection of the many projects that we've done at VCGS over the years. So a bit about, I suppose, the testing or our philosophy here at VCGS. Diagnostic genomic profiling, we think, is a key enabler of diagnostics and also research. So obviously over the years we've gone, the field has gone from many thousands of single gene tests to targeted panels, NGS panels, to exome sequencing, and then most recently up to whole genome sequencing. We've made the decision, I suppose, ourselves that the ability to comprehensively profile the genome of an individual offers quite significant advantages. Comprehensive genome testing is more dynamic. It offers a higher diagnostic yield in first-pass analysis, and also has really high utility for the reanalysis of undiagnosed patients. So at VCGS, we've really moved towards offering sort of exome and genome testing as opposed to targeted panels, which we stopped offering back in 2018. So it's been a little while at this point. And also we've been slowly moving away from our single gene testing as well. So today, exome sequencing is really at the core of many of our testing applications at VCGS. So that goes all the way from diagnostic testing, where we offer comprehensive analysis, which includes all known disease genes, and then plus a gene discovery step where we try and identify novel gene disease associations. We do focused analysis using virtual panels to specific phenotypes. We do offer prenatal testing via exome testing or via exome sequencing, both in the diagnostic setting, in a whole exome setting, but also for targeted variants as well. We do our couple screening using an exome technology, segregation testing, predictive testing in families, and we also do our rapid and urgent testing, or we offer rapid and urgent testing and a reanalysis service. Why have we made this decision to really emphasize the sort of comprehensive, the exome and the genome more so than any other or like smaller sort of more narrow technology? And none of this will be sort of new to anyone. Gene disease associations are not static. Every month, every year, there are new genes associated with disease. There are phenotype expansions for known genes. So a gene that has a known disease association might have a new phenotype associated, a report of a new phenotype associated with it, or a new mode of inheritance. And genes go the other way too. They go from a well-established gene disease association to what was considered an established gene disease association, and they go into the refuted territory where contradictory evidence comes out. And there are numerous organizations around the world that are engaging in this work to really harmonize and establish gene disease associations. ClinGen has a gene curation initiative. The GenCC, the Gene Curation Coalition, aims to harmonize gene disease associations between different groups. And you may or may not be aware of PanelApp England, or the Genomics England PanelApp service, which is the way that Genomics England sources their gene disease associations, and that's a crowdsourced initiative. Here in Australia, we've actually taken the Genomics England approach, and we've sort of forked that and created our own PanelApp Australia out of the Genomics England initiative. So PanelApp Australia is an open online platform for gene and virtual panel curation. We launched it in December of 2019, and at this stage, it has over 250 virtual gene panels in it, sort of separated by phenotype and disease categories. We've harmonized our gene curations between different Australian laboratories around Australia. So this is a public resource, and with Genomics England. And we have more than 100 registered gene reviewers, and this again is open, so anyone from around the world can register to review a gene in PanelApp Australia. We do monthly panel updates where we go through the literature and we identify new gene disease associations. We update panels. Obviously, that still happens in between those big monthly updates, but we do have a dedicated initiative once a month or so to make sure that PanelApp is kept updated. And over the course of 12 months, the last 12 months, we added over 400 new gene disease associations. PanelApp Australia is a member of the Gene Curation Coalition that I just mentioned, and it's hosted locally here at MCRI. We've now got around 6,000 gene disease curations in PanelApp, which is around 1,000 more than OMIM. So it's one of the key advantages we see to this initiative is that OMIM, as good as it is slower to update, I suppose, than something we control ourselves. And so we are sort of outpacing OMIM when it comes to adding those really brand new gene disease associations. We include modes of inheritance for each gene disease associations, and we also have HPO terms attached to our phenotype-specific panels, and that we do monthly updates, as I mentioned. we're adding on the order of 20 to 50 new genes each month. So we've put a lot of effort and resources into our gene curations initiatives and into Panel Australia. And so having a comprehensive genome profiling test, like a whole exome sequencing service, as opposed to a fixed panel, is really advantageous when we want to either go back and look at a patient's data for new gene disease associations, which I'll actually get to in the next few slides, or it also eliminates the need to constantly update panels whenever you identify new gene disease associations. So that's one of the big advantages we see to exome sequencing. And then, as I said, the other is the utility of reanalysis. So we know from the literature that reanalysis increases diagnostic yield or results in new diagnoses. And so there's been lots and lots of studies published. Here we sort of summarized more than 10,000 patients and found a median diagnostic yield of about 15% through over a median time period of reanalysis of 22 months, so just under 2 years. And so the utility of that is obviously high. However, at least in Australia, at least in our service, most negative diagnostic cases are not routinely reanalyzed. And I'd actually go to say that pretty much no negative diagnostic cases are routinely reanalyzed. It's generally done on request only, so a clinician has to come to us and request a reanalysis get done. We know that we get that sort of 15-ish% reanalysis yield, mostly from new knowledge, i.e. new gene disease associations. But that because the reanalysis is generally on request only, there is a substantial lag between knowledge emerging and that reanalysis being triggered. So we have all this data, we have this reanalysis, this exome data. We've already said that exome sequencing, having comprehensive genome profiling, is really useful for the utility for reanalysis purposes. But if we're not doing this on a routine basis, it really diminishes that utility. So we identified several years ago, we identified the need for a regular and automated, in order to reduce the sort of workload implications, reanalysis service or reanalysis pipeline, I suppose, which would incorporate new knowledge and would also minimize false positives in order to ensure the sustainability of that pipeline or that service. Again, there are workload implications here. There's a reason we don't routinely reanalyze negative cases, because it's a lot of work. And so we actually, a few years ago, we received a grant to create an automated reanalysis project or pipeline. And that has results. So that project was led by Professor Zonitza Stark, in collaboration with the Center for Population Genomics, run by Daniel MacArthur and Kaz Simons in the middle, and Matt Welland down the bottom right, were really key in developing this tool. This initiative, or this grant, this project resulted in the creation of a tool we've called Talos. There's a preprint available for Talos now, and what Talos does is interrogates historical exome, again, coming back to that comprehensive genomic data, exome and genome data for new diagnoses. So using a set of sort of strictly defined parameters in order to attain a high signal to noise. So not trying to trawl through the data for every little VUS, but really trying to aim for the clear diagnoses that are just sitting in that data waiting to be pulled out. We've now used that tool to interrogate almost 5,000 undiagnosed research and clinical data sets, and we've got about 248 or just under 250 new diagnoses, for an additional yield of about 5.2%. Lower than the 15% that has come out of those papers, but again, the intention behind this automated pipeline was really to pull out what we call the low-hanging fruit diagnoses, not to pick out every single possible good-going missense variant. This kind of initiative wouldn't have really been as or this project, this pipeline would not have been as successful had we not had many, many years worth of exome and genome data to pull all of these diagnoses out of. So that decision back in 2018 or thereabouts to move all of our diagnostic testing to whole exome sequencing has really facilitated this initiative. And we're in the stages, the latter stages of incorporating that into our routine service that's still to come. But this tool is available to use. It's on GitHub. There should have been a link in the previous slide. And yes, again, that's something that we've seen a lot of value from our exome and genome service from. So I've talked a lot about exomes, obviously, and I've mentioned genomes. Genomes have better diagnostic yield than exomes. That's not really a question that's probably in doubt. There's lots of papers that, well, there is literature out there that makes that looks at that. Here's one example where there were additional variants pulled out of the whole genome sequencing for rare diseases, particularly in the context of deep intronic variants and sort of repeat expansions and so forth, where we know that genome sequencing is better than exome sequencing as a non-targeted assay. So why are we focusing, I guess, so much on exomes? Well, exomes still have key advantages. And again, this is all kind of maybe specific to our service. So this will be in the rare disease setting in the Australian context at VCGS. But we do see that exomes have some key advantages. One of them won't be surprising. It'll be largely economic. Slightly more specific to the Australian system, though. The Australian health system funds some genomic tests. So Medicare is the body that funds pathology testing, amongst other things, in Australia. And as of in the last few years, they've introduced public funding for various genomic tests, the ones that are sort of relevant to us here at VCGS, testing for childhood syndromes, so children under 10 with intellectual disabilities, kidney disease, cystic, non-cystic, and Alport syndrome, mitochondrial disease, and cardiac disease. Now, this is great, obviously, for the Australian public, but the funding that's offered for these indications makes whole genome testing largely not viable, given the current cost of that assay, which really means that whole exome sequencing is the only option if we want to offer this to as a publicly funded test. The second main big advantage that we see for exome sequencing actually comes from the sample types that we're able to use. Here at VCGS, saliva is a really popular sample type for exome testing and for many applications, actually. It has advantages for children, for patients with might have needle phobias, for particularly sort of developmentally delayed children and adults. So a really much less invasive collection process. It allows patients in remote locations to more easily collect samples. They may be quite far away from a collection center, so we can actually ship out a saliva collection kit to them, and they can send it back to us. So that's the self-collection by patients at home, and that also allows telehealth to be much easier if patients can just collect themselves. So you can see from this graph that if we take a look at the kinds of testing our whole exome or our genome sequencing, exome genome sequencing service at VCGS, we receive quite a high proportion of our samples as saliva samples. So if we look at all probands, it's sort of just under 50% or thereabouts, or just over 40%. But if you restrict that to probands less than 10 years old, again, saliva offering pretty good advantages to the testing of young children, particularly those with developmental delay, which is a lot of the testing that we do. You're up over 50% at that point. And interestingly enough, if you we also we offer trio testing. A bit more on that in a sec. And most of the parental samples we receive by far are saliva samples. That's really facilitating the trio testing there. And we've made a fairly dedicated push to increase or to encourage referrers to order trios. I mean, for a variety of reasons. You can see that the proportion of trios we've done since 2020 has been steadily increasing over time. It might have plateaued slightly over the last couple of years, but it has been increasing. There are significant advantages to a trio over a singleton, and by which I mean testing the parents alongside the proband. It makes the analysis more efficient. It reduces turnaround times. We're able to offer a quicker test because we can analyze the sample faster. It can reduce the overall test cost because there's no need for follow-up segregation analysis if you identify a variant and you need to see whether it's in a parent. And because the inheritance information is included all in the one test. We do obviously still offer singleton testing. It's relevant for when we test adults whose parents may not be alive or available. And even in childhood testing, both parents are not always available. So we do offer sort of duo testing with just one parent as well as singleton testing. Being able to offer, from the last graph, you could see that most of by far, the majority of our parental samples are coming into saliva samples. So I think that has really facilitated this push to increasing our trio proportion of the tests that we do that are trios. I'm not sure we would get the same increase if we didn't offer saliva as a collection option. So why am I talking about saliva in the context of exomes versus genomes? Well, we've done some work, and for us, saliva is not suitable for genome sequencing due to the presence of non-human DNA contamination. So we see saliva sample is not a sterile sample like a blood sample should be. There's lots of other things other than human DNA in saliva, notably bacteria and yeast, but also we do get sort of food contamination. So here is an example. It's actually from an exome test because we don't do salivas really from genomes. So what you're seeing here is from an exome. But just to illustrate, even on an exome where we do occasionally see this contamination profile, it can really interfere with the analysis because it looks, for all intents and purposes, like to an analysis tool, unless you go into IGV and you look at the data yourself, it looks like a frameshift variant or an insertion and is often de novo because that contamination may only be present in one of the if you're doing a trio, may only be present in the proband. So you end up looking at a bunch of what end up being false positives. In an exome, it's sort of tolerable. We don't see too much of this. It does pop up every now and then. This specific example is from a Neisseria bacterium. We also see, like I said, yeast, but also cow and chicken we've seen in the past where people have maybe had drunk some milk before they took their saliva swab, not quite following the collection instructions properly. But yeah, this can, in a genome setting, result in a really painful analysis because you end up seeing a lot of what look like high impact variants in genes, but ends up just being because it's not a capture-based test, and it's just sequencing whatever's present in the sample, they're all just artifact calls. So that's the other big advantage we see to exomes. Cost, obviously, in the context of public funding, and also having the availability of this saliva sample type for our exomes. So exomes here at VCGS are here to stay for now. But can we, and we know that genomes are better, they have better diagnostic yield, if you set aside all the sort of cost implications and so forth. But can we narrow the gap with our exome to genomes? And I guess the answer we've gotten to is yes, with an optimized design. And I've sort of highlighted some of the categories of variants that in the paper I mentioned earlier require, they say, genome sequencing. And yes, that's true for the most part, but you can optimize your exome design to capture some of these variant types and in certain situations. So that led us to our collaboration with Twist to develop the VCGS exome design. I'm calling this the version 1 design here. This was several years ago now. So we were using an exome chemistry prior to this, and we moved over to Twist and designed a specific capture for VCGS purposes. And so what that looked like was that we boosted coverage of diet clinically relevant genes, and we also included coverage for known pathogenic non-coding variants. That goes back to the advantage of genome testing being that you could look at intronic variants. Well, yes, but if you can boost your coverage for specific variants that are in introns, if you know that they are pathogenic. And so what we did was we boosted coverage for variants that were pathogenic in ClinVar. And the sort of end result when we compared it to our previous exome that we were using prior to Twist was that we actually ended up with higher quality data, a more relevant design for our clinical application. And we could actually sequence, so less data at 80 times coverage gave better or equal performance to our previous exome at 100 times mean coverage. So we could reduce the cost of our data generation, improve the sort of the economic efficiency of this, but also target more clinically relevant regions. And just to illustrate what I mean by targeting these clinically relevant regions, here's just some screen grabs from a few samples where I'm comparing the previous exome to the Twist version 1, the VCGS Twist version 1 exome. And to show you that where we've increased coverage for these clinically relevant regions. So this is an example of a variant in ClinVar in ANCR-D26, which is multiple times pathogenic in ClinVar. It's associated with thrombocytopenia. It's a UTR variant. There's some coverage down here in our previous exome chemistry, but you can see this really covered quite well in the Twist version 1 exome. Making us confident that we would detect this variant. Similar example here, this is a TMEM216 5' UTR variant. TMEM216 is associated with the Joubert syndrome, Meckel syndrome, and retinitis pigmentosa. And this paper actually describes this UTR variant in individuals of South Asian and African ancestry with retinitis pigmentosa. In our previous exome chemistry is a sort of small amount of coverage here, maybe not reliably detectable, depending on how well the particular sample performs on that chemistry. In the VCGS exome, really well covered. So again, another example of a non-coding variant that you would detect fairly reliably on the updated chemistry, on the Twist VCGS exome chemistry. Some sort of more, so these are the next couple examples are deep intronic variants, where we had no coverage at all in our previous exome, but is covered quite well in the Twist VCGS exome chemistry. This is a sort of well-known, multiple times pathogenic MYBPC3 deep intronic variants, and MYBPC3 is associated with cardiomyopathy and left ventricular non-compaction, so cardiac disease. We do quite a bit of testing here for cardiac disease in adults, so this is very relevant to us. And another example of the same thing, so a deep intronic variant in FKTN that appears to be very common in Korean and Japanese populations in these two publications. This gene is associated with cardiomyopathy and muscular dystrophy, and you can see our previous exome would just not have covered this intronic variant at all, whereas the Twist VCGS exome chemistry has very good coverage here for that variant. Now, obviously, you're not covering every single intron here, but you're really trying to cover those just target the capture to the intronic regions where we know there are pathogenic variants, and we would be really sort of remiss to miss if we can help it. So that was the Twist version 1 chemistry, the Twist VCGS version 1 chemistry. More recently, we developed version 2 of that chemistry, so it retained all of those improvements to clinically relevant regions. Along the way, we've kind of obviously, if you incorporate these known pathogenic ClinVar variants, that's a static set, and over time we've kind of updated the number of the regions for that chemistry. So the V2 chemistry was a sort of more major change to the VCGS Twist exome chemistry. And it had two main improvements over the version 1. The first was for the detection of autosomal dominant polycystic kidney disease. ADPKD is a multi-system disorder that's characterized by kidney cysts, liver cysts, and the increased risk of intracranial aneurysms. It's relatively common for a sort of rare disease at prevalence at birth of 1 in 10,000 or thereabouts. And variants in the PKD1 and PKD2 genes account for about 78% and 15% respectively of cases of ADPKD. Those numbers come from gene reviews. PKD1 and PKD2 encode polycystin 1 and polycystin 2. They're transmembrane proteins. They form a complex or a dimer at the membrane. And so these two genes account for a huge proportion of, or most cases of autosomal dominant PKD. I've mentioned in the previous slide that Medicare, the Australian Public Health System, does fund testing for autosomal dominant polycystic kidney disease from around 2022. But genetic testing for PKD1 in particular has historically been really technically challenging. It's a fairly large gene, but more to the point, it has undergone a number of duplication events over evolution, which means there's a series of pseudogenes with relatively high homology to PKD1, and a lot of NGS chemistries tend to struggle depending on the region of PKD1 you're talking about. Ordinarily, if you're doing sort of single gene testing for PKD1, you're looking at long-range PCR or nested PCR to really get a specific result to avoid those pseudogene issues. There is literature that indicates that whole genome sequencing performs better than whole exome sequencing for the testing of PKD1 in particular, because you have extra read depth in the introns to better map your paired reads to the PKD1 gene rather than its pseudogenes. So that's been sort of a long-standing wisdom with our referrers that genome sequencing is just better than exome sequencing for PKD1, which is true. But what we've done is to really optimize our exome design in this version 2 of the Twist VCGS chemistry to improve the coverage across both PKD1 and PKD2. This improves the detection, of course, of intronic SNVs and indels. It does improve the detection of exonic variants as well, because we get somewhat better mapping, given that we're also improving coverage in the intronic regions. And it also allows us to detect, importantly, structural variants, which do represent a small but definitely not zero percentage of pathogenic variants in PKD1 and 2. Based on this improved coverage, the VCGS Twist exome version 2 approaches whole genome sequencing performance, not quite the same, but approaches it in the PKD1 and PKD2 genes, to the point where we are fairly confident that additional testing by whole genome sequencing of these genes for these indications is expected to have really low additional yield beyond this exome chemistry. And so this is what that looks like visually. The top is the Twist VCGS version 1 exome, which looks very much like a standard exome coverage for a fairly large gene. And the version 2 exome in the middle, which has really boosted coverage across, well, really the entire gene, but looking at the introns there, where there's quite a bit more coverage in here and over here. Um, this is our PCR-free whole genome chemistry, obviously much better, not perfect. It's a complex gene to sequence. It does do better than the exome V2 exome, but the V2 exome is still good enough, again, that we expect whole genome sequencing to have a relatively low additional yield. And so I mentioned that having this improved coverage really facilitates our detection of copy number variants. It does improve our mapping quality across the gene. That's a bit harder to show, but what we've really seen is that these structural variants are really coming up relatively frequently in patients that are referred to us for ADPKD. And so we're getting really good in many cases base pair resolution of these copy number variants, and we're detecting everything from really large CNVs like this one here at around 100 kb, large for an exome anyway. All the way down to single exon deletions, where again you can see we're getting base pair resolution because we have coverage across those introns. And it also throws up some complex structural variants. This is a duplication that's inverted and inserted into a small deleted region here. So you do end up with some sort of sticky examples to try and work out. But again, we're doing the improved. This is all on an exome chemistry, and this is really facilitating our detection, our diagnosis of patients with ADPKD, which is again a fairly given that it's Medicare funded, it's publicly funded, and it's a relatively common-ish disease for a rare genetic disease. We get a significant number of referrals for this indication. And since we've introduced this chemistry, I think in 2022 or thereabouts, we have detected 15 and counting. That was just a number that I was easily able to pull out. Structural variants in the PKD1 and PKD2 genes using this updated assay. Everything from deletions, some duplication there, large multi-exon deletions, smaller single exon deletions. So yeah, really contributing to our diagnostic yield for this cohort. And the final, I mentioned 2 updates we made to our version 2 chemistry. That was the first. This is the second. So the second update we made is to include a recently described disease gene, RNU42, which is a non-coding RNA. It was described in just last year, towards the middle of last year, to explain up to 0.4% of individuals with neurodevelopmental delay. So for a single gene, a huge proportion of cases of neurodevelopmental delay potentially explained by a single gene. All the variants that were detected seemed to cluster in this small little critical region that they highlighted in the paper. And children, neurodevelopmental delay, childhood syndromes, they're a really common referral indication for VCGS. So this is then a really important gene for us to cover if we're doing exome testing for these patients, given that it explains such a relatively huge percentage of cases. Since those papers were published, this is just a screenshot from the ClinVar track in UCSC. There's been quite a few more pathogenic variants reported in that critical region. Obviously, our normal exome capture would not cover this. It's a non-code. It's not a protein coding gene. So our Twist version 1 chemistry didn't cover this. Oh, I should actually mention that just recently there was also a preprint was released showing that potentially there's a recessive disorder associated with this gene as well. So potentially more to come for this gene in terms of how important it is. Yeah, so our previous Twist assay didn't cover that at all. But our new assay, so what we've done with the version 2 exome design is to design probes that cover the RNU42 region. And so you can see that we're getting quite nice coverage of RNU42 now in our Twist version 2 chemistry. Another thing I'd like to highlight with this example is that those papers were published in April and July of last year, and we were able to design and put into service this updated design. So it wasn't in the original design, we had to update it. We were able to update the design and put it into service in October. So we were able to iterate on that design quickly with the Twist assay, which is, I think, a really important thing to highlight. And it means that, so being able to cover this region means that whole genome sequencing will not, going forwards, be required to diagnose that sort of 0.4% of patients with neurodevelopmental delay, if that number holds true. So some final thoughts at the end here. It's hard to argue that whole genome sequencing is the superior technology for obvious reasons. The more even coverage includes all regions, including non-coding regions, improved detection of CNVs and other variant types across the entire genome. We use a PCR-free chemistry here at VCGS for our short-read whole genome sequencing. And we see really good performance from that chemistry, and obviously improvements to come or we are seeing improvements with long-read technologies like Nanopore and PacBio. And because you're covering every single region, you have improved utility for reanalysis and research. It's the more comprehensive of the genome profiles that I mentioned at the start. But exome still remains the first choice for us in routine diagnostic and also in couple screening. It's currently more cost effective than whole genome sequencing, particularly in the context of public funding for these tests. And we can narrow the gap to genomes if we make design improvements to our exome chemistry, which I hope I've demonstrated here. In general, having comprehensive genomic data has many advantages for diagnostic yield and additional value, reanalysis for the driver of new gene disease associations, etc. And as I said, exome versus genomes remain the best value for money for now. That's obviously subject to change over time. Whole genome sequencing is getting cheaper. Long read is becoming more accessible. So eventually that balance will shift, but for now, exomes for us remain the way to go for the bulk of our tests anyway. Yeah, we see excellent value and good diagnostic yield from our exome sequencing technology at the moment.
And once again, thank you and enjoy the rest of your day.